interest group's invitation to me today for the opportunity to give this lecture. Today, I'll be discussing our research characterizing SARS-CoV-2 infection and persistence across the human body and brain among 44 fatal autopsy cases. I have no disclosures. Our study had four primary objectives, and these included to describe the burden of SARS-CoV-2 infection within versus outside of the respiratory tract, to define SARS-CoV-2 cellular tropism across the human body and brain, to determine if SARS-CoV-2 persists in tissues, and if so, over what interval, and then finally to assess if SARS-CoV-2 replicates and or evolves in different anatomic compartments of the body. I'll briefly start by describing our study design. Importantly, we collaborated with the University of Maryland and a total of 7 regional hospitals in the Central Capital and Eastern Shore regions of Maryland that are represented on this slide. Study recruitment was initiated by dedicated upstream clinical partners who often, in the face of surging numbers of critically ill patients, alerted us to COVID-19 deaths for potential participation in this study. Specifically, critical care providers from these hospitals alerted us to patients who had died, and their next of kin expressed a potential interest in study participation. The next of kin were consented by myself, and the remains were transferred to the NIH Clinical Center, which was coordinated through our truly amazing Clinical Center admissions department. And then the following morning, the autopsy and tissue procurement was performed by the National Cancer Institute pathology team in collaboration with my laboratory team, with great effort taken to keep the postmortem interval of our studies within a 24-hour period. We performed a total of 44 autopsies between March 2020 and March 2021. Each autopsy performed is denoted by a red arrow on the Maryland epidemiology curve of COVID-19 cases, which is shown here. As you can see, our cases were well distributed over the first year of the pandemic. It's important for me to emphasize up front that none of these individuals were vaccinated, and all were infected before variants of concern were widely circulating in the United States. We procured tissues from all major organs in all 44 cases, with extensive brain sampling in 11 of these cases. We preserved more than 10,000 specimens for downstream analyses across 85 distinct anatomic sites and body fluids. Pertaining to the results that I'll show in the subsequent slides, fresh tissue and body fluids were preserved in RNA later for downstream analysis by our droplet digital PCR assay for SARS-CoV-2 RNA, for qPCR for subgenomic RNA, which is thought to be a marker of recent viral replication, as well as high throughput single genome amplification and sequencing to compare diversity of SARS-CoV-2 spike gene within and across anatomic compartments. Additionally, flash frozen tissue and fluids were collected for cell culture to evaluate for presence of replication competent virus. And finally, research tissues were formalin fixed for histopathologic analyses, as well as in situ hybridization and immunohistochemistry to query SARS-CoV-2 cellular tropism. I'll now provide you an overview of the demographic and clinical features of our cohort. Our cohort was diverse, with a median age of 62.4 years. 29.5% were female, 41% were Black, 41% white, and 16% were Hispanic or Latino. Pre-existing conditions were prevalent, with 77% of our patients having 2 or more comorbid conditions. Cardiovascular disease, chronic respiratory disease and diabetes, hypertension, and obesity were most prevalent. The median interval from symptom onset to death was 18.5 days, and the median post-mortem interval was 22.2 hours. Most of the cohort were administered vasopressors, antibiotics, steroids, and systemic anticoagulation pre-mortem. 23% were on ECMO, 77% received invasive mechanical ventilation, and 41% received renal replacement therapy prior to death. This section will focus on our characterization of SARS-CoV-2 tissue distribution and persistence. For the purpose of this study, we defined persistence as detection of raw RNA in tissues greater than or equal to 31 days after initial COVID-19 symptom onset. For ease of presentation, I'm going to show a heat map depicting our SARS-CoV-2 ddPCR RNA results, limited only to the 11 patients from whom we collected both body and brain tissues. To orient you, on the left side of this figure are the tissues we sampled, organized by tissue groups. These groups included respiratory, cardiovascular, lymphoid, gastrointestinal, genital urinary, endocrine, skeletal muscle, peripheral nervous, and ocular tissues. Each column represents a patient with their ID number listed at the top, and denoted at the bottom is their duration of illness prior to death. Illness duration is grouped into early, mid, and late cases, defined by death within 14 days, between 15 to 30 days, or greater than or equal to 31 days following symptom onset, respectively. Dark red represents SARS-CoV-2 nucleocapsid copy numbers, or high numbers of SARS-CoV-2 nucleocapsid, and dark blue represents low copy numbers by our ddPCR assay. As you can see, we detected SARS-CoV-2 RNA across all tissue groups, with the highest levels detected in respiratory and cardiovascular tissues among the early group. Additionally, we observed relatively high levels of SARS-CoV-2 RNA in P33, or patient 33, in the late group at 76 days after illness onset, and notably detected SARS-CoV-2 RNA in multiple tissues in P42, shown all the way to the right, who died approximately 8 months after COVID-19 illness onset. In this particular case, the patient recorded only mild COVID-19 illness from which they recovered, but months later contracted hepatitis A that resulted in fulminant hepatic failure that led to liver transplant and ultimately death. Displayed here are the ddPCR results for the same patients across 10 distinct anatomic sites within the central nervous system. We detected raw RNA in all regions tested and detected residual RNA in all 6 late cases, boxed in red, including across multiple regions of the brain in P42, again all the way to the right, at 230 days after initial infection. In order to more fully characterize trends in SARS-CoV-2 RNA burden within versus outside of the respiratory tract, we performed a linear regression of RNA levels in respiratory versus non-respiratory tissues by illness day. On this figure, log transformed RNA levels are shown on the y-axis and day of illness is on the x-axis. We observed that SARS-CoV-2 RNA levels were significantly higher in respiratory tissues, indicated by the red color, compared with non-respiratory tissues, which are shown in blue, for months following illness onset. A significantly steeper rate of decline in raw RNA levels, or negative slope, was observed in respiratory compared with non-respiratory tissues. The y-intercept and slope values for both grouped and individual respiratory and non-respiratory tissues are summarized in the table to the right. We also performed a linear mixed model analysis of RNA burden in respiratory versus non-respiratory tissues in our predetermined early, mid, and late groups. Among the early group, we observed approximately 100-fold higher SARS-CoV-2 RNA levels in respiratory compared with non-respiratory tissues. A 20-fold and 4-fold difference were observed in the mid and late groups, respectively. Here's the same heat map that we looked at before, but now with the results from the qPCR evaluating for subgenomic RNA, again, a marker of recent viral replication, denoted by vertical hash lines overlying our color-coded heat map. I'd like to first highlight that the 4 earliest patients, representing a duration of up to 18 days post-illness onset, all had subgenomic RNA detected across most respiratory and many non-respiratory tissues, including the brain. We also detected subgenomic RNA sporadically in some of the late cases, including across 4 different tissue groups in P33 at 76 days following illness onset. To better characterize the relationship between SARS-CoV-2 RNA levels detected by our ddPCR assay and levels of SARS-CoV-2 subgenomic RNA detected by qPCR, we performed a number of Spearman correlations. Among the 10,025 samples that were ddPCR positive and also evaluated by qPCR for subgenomic RNA, we observed that the RNA levels between the two assays significantly correlated with a correlation coefficient of 0.76. This association strengthened to 0.88 and 0.86 when the analyses were restricted to the 496 samples from early cases or the 396 samples collected from respiratory tissues, respectively. When the analysis was limited to the 302 samples that were positive for both of our assays, a significant correlation coefficient of 0.91 was observed. Finally, we performed a regression analysis using the quantity of RNA detected by ddPCR to predict whether a sample would be positive for subgenomic RNA. We found that a ddPCR value of 1.47 copies per nanogram of RNA input into our assay predicted a positive subgenomic RNA result with 93% sensitivity, 91.6% specificity, with a receiver operating characteristic, or ROC, area under the curve, or AUC, of 0.965, supporting the strong predictive capacity of this model. After detecting subgenomic RNA in over 300 samples, we sought to determine if we could isolate SARS-CoV-2 in vero cell culture. We attempted to isolate SARS-CoV-2 from 55 respiratory and non-respiratory tissues across 20 patients. We successfully isolated virus from 25 tissues collected from 9 patients who died within 1 to 12 days of illness onset. In these tissues, cytopathic effect and an increase in SARS-CoV-2 subgenomic RNA levels and cell culture supernatant relative to levels in the original tissue homogenate were observed, confirming replication-competent virus. We further characterized the relationship between subgenomic RNA and replication-competent virus and observed that 100% of tissues with a subgenomic RNA cycle threshold between 15 and 20 had replication-competent virus. This number decreased to 64.3% of tissues that had a CT value between 20 and 25, and further decreased to 31.1% of tissues that had a CT value between 25 and 30, and no replication-competent virus was detected in tissues above a subgenomic RNA cycle threshold of 30 or higher. Overall, we're able to isolate SARS-CoV-2 from nasal placode, sinus turbinate, bronchus, lung, and also in the non-respiratory compartment, the choroid sclera of the eye, the jejunum, the mediastinal lymph node, the heart, and the adrenal gland. We sought to estimate SARS-CoV-2 RNA levels that were strongly predictive of replication-competent virus in vero cell culture. To accomplish this, we performed regression analyses with associated ROC curves and found that a ddPCR value of 758 copies per nanogram of RNA inputted into the assay predicted replication-competent virus with a 76% sensitivity, 90% specificity, and an ROC-AUC of 0.887. A subgenomic RNA value of 25,069 copies per microliter of RNA predicted replication-competent virus with 72% sensitivity and 100% specificity with an ROC-AUC of 0.915. These analyses provide a robust framework to predict a tissue run on ddPCR and or subgenomic RNA qPCR to determine if it is likely to have replication-competent virus without having to perform cell culture in a BSL-3 setting, which is obviously a limited resource. Now I'll briefly take you through our in situ hybridization results characterizing SARS-CoV cellular tropism in our cohort. Using our ddPCR and subgenomic RNA qPCR results as a guide, we utilized an in situ hybridization probe probing the spike region of the SARS-CoV-2 genome to validate our PCR assays and to determine cell type specificity within tissues. While not shown here, positive and negative controls for a human housekeeping gene and an E. coli gene were assessed to support specificity of this assay. While we found positive staining in all tissue groups, I have a small panel here that highlights the ISH staining in brain and other tissues. Displayed in the first row, in order from left to right, are frontal lobe of a left case A, corpus callosum of an early case B, cerebellum of a late case C, and cervical spinal cord of a late case D. Shown in the second row are follicular cells of the thyroid, stratified squamous epithelium of the esophagus, as well as the stroma of a capillary with the number symbol there. Mononuclear leukocytes within the white pulp of the spleen, which is panel G, and colonic epithelium, same panel. And mononuclear, spleen is G, and in H, colonic epithelium by the asterisk, and mononuclear cells within the stroma of the appendix, which is the pound symbol there. In the final row, the ISH staining occurs within the endocrine secretory cells of the adrenal gland, that's I, the stromal cells of the postmenopausal ovary, J, Sertoli cells and maturing germ cells in the seminiferous tubule of the testes, that's K, and the endometrial gland, epithelium, and stromal cells of a premenopausal uterus in L. We further evaluated SARS-CoV-2 cellular tropism within the cerebellum and other regions of the brain via fluorescent immunohistochemistry and confocal microscopy. In these images of cerebellum, cell nuclei are pseudocolored blue, SARS-CoV nucleocapsid is green, and neurons are magenta. The locations of the molecular layer, denoted ML, granular layer, GL, and white matter are indicated on the slide. Panel A shows a low magnification visualization of a no antibody control. And B shows an adjacent section with viral-specific protein expression. Higher magnification of images of B, shown in C through E, demonstrates cell type specific infection. In C, positive neurons are indicated by the yellow arrows. Other unidentified cells indicated by the white arrows are also associated with viral protein in the granular layer. In D, Purkinje cells adjacent to the molecular layer are infected. And in E, in rare instances, blood vessels adjacent to the granular layer and white matter were associated with viral protein. This table summarizes the 36 cell types and hyaline membranes across the body and brain that were ISH positive for SARS-CoV-2 spike RNA in our cohort. You may recall that our final study aim was to determine if SARS-CoV-2 evolves in different anatomic compartments within the same patient. To address this question, the Boritz lab performed high throughput single genome sequencing to analyze spike gene variant sequences from a total of 46 tissues from 6 individuals. Sequences from P18 or patient 18 are shown on the left, which were identical across all anatomic compartments tested. P19 on the right, in this patient we detected a single synonymous mutation, which is denoted in blue here, in a minor variant in the right superior lung lobe. Overall, these two patients demonstrate that SARS-CoV-2 can infect cells within non-respiratory tissues without requiring changes in the spike genome. In patient 27, which is shown on the left, synonymous mutations were detected both within the respiratory tract and outside of it, specifically in the mediastinal lymph node and the left and right ventricle of the heart. And again, those synonymous mutations are denoted by blue there. While we were unable to sequence many tissues from P33, who was a late case, shown on the right, there were 2 distinct haplotypes detected within the thoracic lymph node of this individual. Finally, we were able to sequence brain samples from 2 patients in our cohort, which are shown here, which revealed non-synonymous mutations between haplotypes within the CNS and the rest of the body, specifically in the dura mater for P36, which is shown here, and the hypothalamus and the thalamus of P38, which are shown here. These results indicate that SARS-CoV-2 within the brain is subject to the development of genetic compartmentalization, and exploring the mechanisms behind this will be important future work. I'll now briefly summarize the histopathologic correlates of SARS-CoV-2 infection in our cohort. Our pathology colleagues in NCI, in addition to running all of our ISH assays, determined the cause of death and characterized the histopathologic findings for all cases. 86% of cases died from COVID, while the remainder died with, but not from COVID. Diffuse alveolar damage was detected in all but 18% of cases. A panel shows panel A on the right shows the exudative phase of diffuse alveolar damage with hyaline membranes and mild interstitial inflammation from an early case, and panel B shows the proliferative phase of DID with sparse inflammation in a mild case. Excuse me, not mild, a mid case. Acute pneumonia was present in 61%, and pulmonary hemorrhage was detected in 32%, demonstrated diffusely in panel D of the images on the right, and pulmonary thromboembolism, was detected in 23%, an example of which is in panel C in a medium vessel, pulmonary vessel on the right. That's panel C. We're able to quantify the temporal progression of diffuse alveolar damage from the early exudative phase, which are shown by the orange stacked bars during week 1 to 2, to the intermediate proliferative phase, which are the gray stacked bars, into the late fibrosing phase after 1 month of illness, which are shown by the yellow stacked bars on this figure. In the heart, we observed myocardial infiltrates in only 9% of our cases, despite detection of viral RNA in the heart of 80% of our cases. Patient 3, which is shown in panel E, is an example of a single case that had active lymphocytic myocarditis at the time of death. Bland acute microscopic ischemia and myocardial necrosis was observed in 9% of cases, an example of which is shown in panel F of P38 on the right. We evaluated for the presence of lymphodepletion in lymph nodes and spleen among all cases. Lymph node architecture remained intact without lymphodepletion among 78% of cases. Panels M through O on the right show lymph node of P25, an early case, which is well-defined follicles with marked plasmacytosis and histiocytosis. In contrast, some degree of lymphodepletion was detected in the spleen in all but 5% of cases. Panel K is an example of lymphodepletion in the spleen compared to relative preservation of the white pulp shown in panel J. We found no evidence of direct viral cytopathic effect in the brain, despite detection of viral RNA in 10 of 11 or 91% of our cases. Instead, we observed nonspecific findings, including focal or diffuse hypoxic ischemic injury in 45% of cases. Vascular congestion was observed in 45% of cases, and microscopic hemorrhage was observed in 18% of cases. In summary, we observed that SARS-CoV-2 is widely disseminated across the human body and brain early in infection, that SARS-CoV-2 RNA can persist in multiple anatomic compartments for over 7 months, and we observed a paucity of inflammation or direct viral cytopathology outside of the lungs. With that, I'm going to take a little bit of time to make many important acknowledgements. I'd like to start with the many healthcare providers at our consortium hospitals that are listed here. I'd like to specifically acknowledge Dr. Allison Grazioli, who spearheaded the provider engagement and patient enrollment at all of our consortium hospitals, as well as to acknowledge Drs. Kapil Saharia and Joe Rabin, who have been partners in multiple downstream projects related to this cohort. This work would not have been possible without the enthusiastic participation, leadership, and support provided by Drs. Hewitt, who performed our ISH assays, and Dr. Kleiner, as well as the staff and trainees within the NCI Laboratory of Pathology. Drs. Emmy DeWitt and Vincent Munster and their laboratory members supported the subgenomic RNA and viral culture work that I showed. Drs. Karen Peterson and Clayton Winkler led the fluorescent immunohistochemistry work in the brain. Dr. Bortz's group performed our spike sequencing. And of course, I acknowledge and recognize Dr. Holland and the intramural program for both encouragement and funding to support these studies early on and continually through this pandemic. Many members of the critical care medicine department contributed to this work, directly or indirectly. I'd like to specifically acknowledge Dr. Sidney Stein, who's the first author of this work, and Sabrina Romelli, who worked together to perform the ddPCR on thousands of our tissue specimens. I'd like to also thank Dr. Junfeng Sung in our statistics support, who performed all of our regression analyses. Finally, I'd like to acknowledge and thank Tracy Lewis and all members of the NIH Clinical Center Admissions Department staff for their agility and adaptability in supporting our requests for transfers of next of kin or deceased, often in the wee hours of the morning during this 1-year interval, over and over again. And with that, I will thank you and close and take questions.
Okay, Dan, thank you so much for a very interesting talk. We have a lot of questions, but before I start with them, I just want to reiterate the CME code number. For those of you taking this credit, it's 37971. 37971. Okay. So I'll start with a question from Jim Boyce. Do post-mortem analysis of people who've had mild COVID-19 and who have recovered from it and died of something else after, do they show the same cellular tropism and or persistence of SARS-CoV-2?
Excellent question, Jim. We're very cautious and openly acknowledge that our cohort was largely limited to older individuals with a number of comorbid conditions that develop severe and ultimately fatal COVID. And so we're cautious about generalizing our observations to a younger, healthier population and or including a population that's been vaccinated or infected with variants of concern. So those are all limitations. Having said that, there are a small number of individuals in our cohort that were young. We had a 6-year-old who presented to the hospital for a complication of a seizure disorder unrelated to COVID and was incidentally found to have COVID, where we did find the same distribution of virus across that individual's body. And of course, I mentioned the one case that was in an adult where this person had reported mild respiratory symptoms and then 8 months later, unrelated to COVID, as far as we know, developed hepatitis A infection through foodborne illness and died of an unrelated complication. And that person, 230 days after their initial illness, we obviously found persistence of viral RNA in many compartments. So the short answer is we're careful about generalizing our observation to a different population, and we do think that those studies need to be done.
So this is kind of connected to Karana's Surender's question. He's asking whether viral RNA histopath findings are minimal lower in late cases. What's the cause of death in those late cases? So it's
Yeah,
All
so
right.
Surinder, in the short answer is in our preprint, we have a table that summarizes individual level patient data that will specifically outline the cause of death for each patient. But in general, those individuals that I mentioned were older and had more comorbidities. More often than not, were admitted to the hospital, remained in the hospital for an extended period of time, were provided life support, and died due to COVID complications, typically of respiratory failure. And in that group, we obviously have then, a nice timeline of when their symptoms started. They were observed in the hospital for an extended period of time, and then we have the interval from at the time of death. As I mentioned, a really interesting finding that I think that we're focused on, and I think it needs to be focused on, is, why is it that the virus gets to remain in plain sight in all of these extrapulmonary sites in the absence of histopathologic evidence, largely histopathologic evidence of injury and or inflammation? How is it that the virus is eluding the immune response in those extra respiratory sites?
Quick question from me before I get to the next one. The patients where you saw brain infiltration, where, for instance, your Purkinje cell staining, it wasn't clear to me, was that a structural staining to a structural protein of the virus, or was that actually indicating low-level replication in Purkinje cells? Could you?
So the target for our immunohistochemistry was a nucleocapsid protein.
Okay.
So that was nucleocapsid. Based upon, we also, confirmatory assay by ISH. So we approached the brain with a number of additional assays because obviously there are major implications about finding virus in multiple regions of brain and specific cell types. So we're quite cautious about those observations and have taken a number of assays to support or confirm that cell type specificity. As far as replication in the brain, Nihal, we have right now indirect and are pursuing direct evidence of replication competent virus in the brain. The indirect evidence is that the sequencing results that I showed you show evidence of minor variants in the brain, and sometimes not minor variants. Actually, they're nearing equal distribution of variants with non-synonymous mutations that we detected in the brain that we did not detect in the respiratory tissue, suggesting that ongoing viral replication does take place in the brain and that there actually is some evolution of the virus in the brain. We have tried to grow virus from the brain. We're not done. We're going to persist. So far, we have not definitively accomplished that, but it is something that we continue to pursue.
And your answer is related to two questions. So let me ask those. One is from Roland Owens. Do your virus distribution studies suggest any novel receptors? So, for instance, in the brain, what's the distribution of the classical receptors versus what might be novel? And then second question is from Carolyn Ugaru. Thank you for this extremely insightful seminar. Can you please briefly elaborate on the genetic compartmentalization you see in the infected brain tissues? What exactly does that mean? You kind of alluded to that, so maybe you can reiterate and give
Yeah, that's perfect. Yeah, Dr. Owens, thank you for your question and also for the second question as well. Specifically, we did not or have not yet queried the association with the known SARS-CoV-2 receptors and co-receptors in tissues and co-localization or presence of a virus by our assays, largely because others have focused on the proteome map of the human body and presence or absence of these receptors in different tissues. And so we didn't pursue that. Obviously, that is something that we could pursue. You know, we cannot directly answer that question, but there are cell types that are not known to have TMPRSS2 or ACE2 receptors. Monocytes are an example, and this has been now recently published in Nature, as I think Nihal realized, she's shaking her head, that, you know, there's evidence that those cells are either infected or perhaps take up opsonized virus and are able to accomplish that in the absence of the traditional SARS-CoV-2 receptors. So I think that's work that we can pursue, and that's additional work that likely needs to be accomplished. As far as the compartmentalization of the virus, I do think I showed 6 cases, and I think perhaps the last 2 cases, which are the 2 cases where we had viral RNA that we could query, not just from non-CNS sites, but also from the brain. In those 2 cases, and I guess I could bring them up, but anyway, in those 2 cases, the important observation, and again, this is an N of 2. I think it's something that needs to be followed up with larger cohorts and cohorts that sort of emulate our study design, that sort of optimized tissue preservation and short postmortem intervals to be able to glean the observations that we've had. But the observation in an N of 2 is that there were viral variants in the brain, populations of viral variants in the brain that were not detected in the respiratory tract, suggesting that there's seeding of a virus in the brain, there's ongoing replication in the brain, and that we obviously know there's an RNA virus that's error-prone, and that as it replicates, there are variant populations that are produced, and through the very sensitive sequencing technique out of the Boritz lab, we were able to detect variants in the brain that were not detected in the respiratory tract, suggesting that there is replication there, and perhaps that there's a particular immune environment there, and this is speculative. Obviously, that's an immune protected site that would allow for, you know, viral replication and evolution of the virus.
And related to that, again, there's a lot of questions about the brain, naturally. In the CSF, were you able to find any viral genomes and antibody levels? Were you able to quantify it all in the CSF?
Yeah, good question. The short answer is I don't know. I need to phone a friend from Dr. Stein. You know, so I know as to whether or not we collected CSF. If we collected CSF, it was on a limited number of individuals, and whether or not we detected viral RNA or subgenomic RNA, I cannot recall. I'd have to phone a friend and check our preprint. But what I do know is that we didn't pursue evaluation of antibody responses, or, you know, for example, soluble mediator responses or otherwise in CSF. I'd have to go back to review the inventory to determine what exists that might be accomplished with that sample type.
Sticking with the CNS, have you seen the presence of viral particles in eye tissues? From Ramakrishna is asking.
Yeah, thank you for that question. So we did query eye tissues. We found presence of viral RNA in eye tissues. That is summarized and categorized in the preprint. What's not in the preprint are confirmatory characterizing cell types in the eyes, and that may or may not be forthcoming in a separate body of work.
This is Thomas Sabotka asked this question, but is there any indication? I mean, this is very speculative, that residual COVID-2 in the brain or other tissues could become reactivated to produce some type of disease condition in the future. I mean, I think it's very early to say, right? I mean, we're at the beginning of this.
So you're correct, Nihal. That is your answer is the correct answer. We can't answer that based upon the study that we did. It wasn't designed as such, but for certain, those are the types of questions that need to be pursued in the future. And obviously, there are concerns or questions about, well, what is the relationship between, post-acute sequelae of COVID or PASC, and our observations of persistence of viral RNA in many anatomic sites. And again, acknowledging a limitation of our study, we didn't query a PASC population. And so we're cautious about making overstatements about how our findings relate to PASCs. But what it does highlight is that in that patient population, that there should be studies that are designed to specifically evaluate whether or not there is persistence of viral RNA in different anatomic sites, and to what degree that persistence is associated with the clinical manifestations of PASCs.
Thank you. And there's a question from Chase Nelson, I guess relying on what you know about the literature. How likely is it that other respiratory viruses, say common cold coronaviruses, also have similarly widespread tropism and persistence in non-respiratory tissues? Or has it, been gone undetected? It's there, but
yeah, that's a great question, and it's one that we have had some really enjoyable conversations and debates around. And the implications are, should we extend this approach to fill in the blank X, Y, and Z respiratory or other viral illnesses that are associated with human severe human illness. What I'll say is this, that part of our approach for COV-2 was informed by the historic literature of COV-1, same receptor, MERS, different receptor, a smaller body of literature, but a literature that fairly clearly defines that these coronaviruses, at least the severe human coronaviruses, make their way outside the respiratory tract and infect tissues elsewhere. And so based upon that literature and our sort of understanding, we pursued this quote unquote aggressive approach to better understand COV-2. If you look at the literature, for example, influenza, I don't think that folks have taken the type of approach that we have to characterize influenza viral distribution across the body and brain. But having said that, people have been studying influenza for a really, really long time and have done a lot of careful studies. And those studies do suggest that the prevailing knowledge and dogma is that those viruses, at least the predominance of them, absent the high-path avian influenza viruses that have this special polybasic cleavage site that allow them to get outside of the respiratory tract, that the seasonal influenza viruses that we're infected with don't do this. But of course, if you don't look, you don't know. And so those are ongoing discussions. We're happy to be interested in other people's thoughts, but we are discussing those among ourselves.
And certainly there's none of this anecdotal, at least clinical neurological, evidence that people say they have with, even personally, speaking from my, having experienced COVID personally, the brain fog, the vertigo, these things persisted for months after I, tested negative for the virus. So none of those you don't hear from patients with flu or the common cold. It's too many right now that it suggests, really.
It's really tough, Neil. It's super tough because, there's a literature out there in the non-respiratory complications of flu, but most of them appear to be, not due to direct viral infection in those organs, but due to indirect effects. And you're right, the PAX is a major issue, and now there are, strong data supporting that this is not just a vague constellation of things, that it is related to prior infection. And so beginning to unravel the pathogenesis of that, I think is, and obviously the NIH has, I think, devoted over $1 billion in research funding to that particular question.
Right. And can you comment on this recent study in Nature where they had followed these patients with fMRI and seen sort of shrinkage of gray matter? How does that connect?
I love that study. I think that study is fascinating. strongly suggestive, right? I mean, it's really that study from the UK took advantage of these individuals that were systematically studied by MRI pre-COVID era, and then had the ability to follow the same people post-infection and correlate them with specific functional MRI regions that were really, really fascinating, and found a strong signal that something's not right. And whether that something is not right, persistence of viral RNA in the brain is contributing to that or not, I think that's something we got to figure out.
Right? I mean, it's so important, so important. There's a question about the sensitivity of your infection assays when you're harvesting virus. Could it be that you're missing out on virus levels because your sensitivity is not there in those ex vivo infection assays?
The short answer is absolutely yes. And that is even though that's a simple answer, it's a hard message to translate to the average listener. We know that cell culture is an insensitive way of detecting replication competent virus, and but it's the standard way, and it's the gold standard way. And so the way to get around this, or to try to quote unquote increase sensitivity to find replication competent virus in samples that didn't grow in cell culture, is you can manipulate your cell culture. You can put in humanized receptors. You can take it into a different system. In other viruses, as you well know, Nihal, people have used immunocompromised mice that are neonatal mice that perhaps are particularly susceptible to viral. So there's other ways but viral culture is an insensitive way of detecting replication competent virus. And so is there likely more than we're observing and claiming? Probably, but we have to work with what we have.
There's a question from John Sinew. I wonder if you looked into what percentage of people who were vaccinated had persistence in the brain. Did you have any?
Again, I was careful, and I'll reiterate, to acknowledge that a limitation of our study is that this was done during the first year of the pandemic, before availability of vaccines, and none of the members of our cohort were vaccinated. We know, as I know the person who asked the question knows, that vaccines are remarkable as far as preventing severe disease and death. How good are they at preventing viral dissemination to extrapulmonary sites? Don't know. I would hope and think that they're probably pretty good, but again, would be a question worth asking. We know that, for example, and again, not to confound issues here, that even vaccinated people that have breakthrough infections that go on to experience, symptoms of PAX, that they can, mild illness, you can have it, post-vaccine breakthrough infections, you can have it. Again, I'm not trying to confound those things. I'm just identifying questions that remain to be answered, but I think that is a question that remains to be answered.
Let's see. Howard Young is asking if there was an interferon signature in the brain.
Howard, you ask a very good question. And the answer is we don't know yet. That's on the list of future work. And to be specific, this first effort was really focused on the pathogen, as you noticed. Our group and our number of collaborators, both within NIAID and many other intramural institutes, as well as a number of extramural collaborators, are beginning to shift our focus to apply various technologies to focus on, more on the host response beyond, histopathologic evidence or evidence of inflammatory cells in tissues. And one of those studies is directly focused on cellular responses in the brain using available tissues. One of the benefits of collecting flash frozen specimens that are immediately adjacent to specimens where we've characterized, in great detail, RNA levels by ddPCR and presence of RNA in different cell types by ISH, is that those flash frozen samples are amenable to technologies like single nuclei seq, and then obviously application of additional technologies where for example, spatial transcriptomics and those types of things to begin to answer that type of question. Others have applied those tools in the brain and elsewhere, we do intend to pursue these things in our cohort.
Okay, well, thank you so much, Dan. It's been very enlightening, and this ends today's seminar. Thank you.
Thank you, Neil. Thanks, everybody.